Good afternoon, everyone. My name is Sam, and today I'm excited to talk to you about how we're going to build a more sustainable future using biological chemistries. Many of you may know that our dependence on fossil fuels is releasing billions of tons of carbon dioxide and other greenhouse gases into the atmosphere every year and is a major cause of climate change. What you may not realize, however, is that more than half of every barrel of oil that we refine goes into making other products other than fuel. And so when we think about how we're going to build a more sustainable future, how we're going to wean ourselves off of dependence of these fossil fuels, we need to also think about how we're going to replace these chemicals that are in everything from crayons to paint to shampoo to aspirin. And so what I might challenge, or I guess the question we're asking is, how can we build these technologies? How can we find new chemistries that allow us to accomplish these technological challenges and build these products that we all depend on? What I might propose is we could take a leaf out of nature, because biology and nature has been doing this for billions of years. Biology has built this beautiful and complex and efficient world that has built such an amazing world that we all depend on. And it does this by kind of building function from the level of DNA. So could we understand how biology works and reverse engineer technologies using those same principles? As a synthetic biologist, I'm really interested in answering those questions and taking our understanding of life down to the level of DNA so that we can build back up from that scale. So if we look at this leaf, we can kind of see how this complex veined structure contributes both to nutrient transport in and out of the leaf, as well as structural support to kind of provide a solid foundation for that leaf to be stable in the tree, as well as kind of having a green color and many different machines within each cell in that leaf to carry out photosynthesis, and also kind of having these specialized organs on the underside of the leaf to allow water to evaporate and help to cool the entire organism. When we think about how life does this, it really builds from the level of DNA. And thinking about these link scales involved from the level of DNA, cells use that DNA to organize themselves into more complex structures. And then from those cells, organisms will build tissues and organs from those tissues, and then systems, and then going into the organism. And then even those organisms are kind of built into communities and ecosystems, and finally the entire biosphere. And what I'm doing is actually focusing on that very fundamental level at the level of DNA so that we can ask these questions of how can we build this function in bacteria specifically. So how does an E. coli cell use its DNA? So starting from its genome, which is about 4.5 million base pairs, or 4.5 million A's, C's, G's, and T's, it will take, it's about 4,500 genes, and make a copy of those genes when it wants to make the product from them. And then from that copy, it'll send that copy to this system called the translation system, which makes what's called a protein from it. These proteins are incredible and diverse and amazing molecules, and this process actually is so essential to cells that it's called the central dogma of molecular biology. Every single cell in the world depends on this very fundamentally. Why are these proteins important? It turns out that they're the labor force of the cell. This is how the cell accomplishes all of its functions. It makes a protein for controlling the flow of water into the cells. So this protein will sit in your cell membrane and control water molecules moving in and out of the membrane by forming a pore in the membrane. There's a protein that will harvest carbon dioxide from the air in the process of photosynthesis. There's a protein that transports oxygen throughout your body. But also, proteins are useful products for human use. We already make and manufacture many different kinds of proteins for human use, such as chymosin, which is used in cheese fermentation, such as insulin, which is important for diabetics, and such as amylase, which is an enzyme that goes into your laundry detergent. In our lab, we're really interested in actually engineering the process of protein making, this process of translation itself, to be able to expand the chemistry that we can put into proteins and make new kinds of products, expanding the range of useful products that we can make from these biological systems. So how would you do this? It turns out that translation process that I was talking to you about before is so essential to the cells that we can't really interrupt it, we can't mess with it, because it very quickly leads to cell death. So what we do instead is try to split that central dogma of molecular biology into two pathways, one that's still supporting the cells, that's making all the proteins that it needs to function, and one where we kind of siphon off some of the genes that we actually want to make these new kinds of proteins with into a pathway that we call orthogonal translation. where we set up these engineered factors that we've made in the lab and make these new kinds of proteins that we're wanting to make. But I have no clue how to make a cell that actually does this well. I don't think anybody does because this is a very complex problem. When you look at the genome, it's just 4.6 million different letters, and I can't make any sense out of that myself. I really don't know how to build a good system, and I don't think anybody does. We need to figure out how to encode this complex translation system into the genome in order to solve this problem, in order to make new kinds of proteins that we want in these cells. And this translation system, the native translation system, actually contains over 100 different factors that all kind of come together in a beautifully coordinated and well-oiled machine, basically, to make these proteins. And so when we think about building something of similar complexity, it's not only about getting the parts inside of the cell, it's about getting them to work together well. And we don't really have any good way to know how to do that. So instead, what I'm trying to do is build a genome engineering platform where I'm shuffling DNA inside of cells and generating a bunch of diversity in a population of bacteria. So I can very easily generate a million or 100 million bacterial cells within one culture. And then I can use this process that I'm building to shuffle the DNA inside those cells. And then I can kind of borrow a trick from nature and try to evolve those cells so that I can generate and select on that diversity that I'm making in the genome. So from that culture, I can make potentially over 1 million different kind of diverse cells, and then find the ones using a selection platform to isolate new healthy cell lines that are going to be these new chassis strains that we use to build these new kinds of proteins. And so scientifically, I would want to conclude by talking about just, I'm excited to work on building a better world from DNA. I think there's quite a lot of potential to use and harness the potential in biology to make new kinds of materials and medicines. And by building better genome engineering tools, I'm hoping to expand the kind of challenges that we can address in biotechnology and kind of allow us to have new tools in our toolbox to make new kinds of technologies that will be useful for the world. These are a few resources that I'd like to just kind of highlight real quickly, just so that if you're interested in learning more about synthetic biology and where the field's going, you can kind of look through these for some background. So you should be able to find these on our website after this is posted. And finally, I want to thank my lab and especially my advisor, Mike Jewett. Thank you.